This is kind of presenting an idea and some preliminary data that I think is really promising. I have very few data slides. One. But I hope it gives you the idea of what we're doing and why it's exciting.
Is this one right?
Yes. Thank you.
Excellent.
So this is work that was done by myself and Yannick Malik. Yannick had the accepted talk here and was unable to come. Luckily, I am also on the project, leading this project. So this is really kind of so I'm from Pacific Northwest National Laboratory in Richland, Washington, this is really drawing on our deep work in mass spec assisted omics, proteomics and metabolomics specifically. And I'll try to explain this fairly quickly. So this kind of audience knows these, this kind of slide, which is like, there's a lot of unknown stuff, right? Protein dark matter, we variously call it different things. Some 30 to 60% of proteins, even in well-characterized organisms, have either no annotation or not very useful annotations. And how do we get in further to that space? The kind of traditional method, and I think no one should be that surprised about this slide either, is that we use associations. And the associations that we commonly think of for doing this kind of mapping are based on sequence similarity, where we can infer homology. We heard some really nice talks about that earlier. And then we take a known experimentally derived annotation and we say the homolog must have that same function. If you were at Mark's talk earlier, that casts a whole different light on whether or not that, in some cases, that is an appropriate thing to do. This is how things are done. And protein language models and to a certain extent structure models that are predicted are also based on the same idea, right? Like you're leveraging the sequence to get at these associations. And we've developed methods. I think I spoke last year at ISMB in part on this. This is a very simple K-mer-based approach where we throw a few tricks in. It's very fast. We've gotten very good performance out of it, and it's kind of flexible and fine-tunable. I'm not going to focus very much on this. This is just application to a large metagenomic data set. And we're showing here that we're getting some really interesting annotations that aren't being gotten by some of the more conservative methods. We're still working out some of the details and making sure that this is all real and not kind of hallucinated in the old school sense, where we're squinting our eyes before we kind of put this out for publication. We were interested in other ways of mapping protein function. And again, I mentioned that we have large work at PNNL on mass spec assisted omics. I'm not going to go into the details of how that's all done and what kind of data, but essentially you got a matrix out. And this is representing two different matrices. From on the left-hand side, the protein mass spec data. The rows are different conditions. So these could be different culture conditions. They could be different environmental conditions. They could be different mutations. And the columns equal, different proteins that you've identified. Now, these columns should be thousands long because we identify thousands of different proteins. And then on the right, we have metabolites. So these are small molecule metabolites, which we identify by mass spec. And again, you might get thousands of those out there. And we do this on matched data. So we have published data sets and we have data sets that we're generating that basically we've cultured and isolated the same samples, and then we split the samples and we do proteomics and metabolomics and sometimes other kinds of omics on these. I'm choosing proteomics and metabolomics because it's kind of a novel application. And I think there's some real room for kind of getting more information out that way. So the real heart of our idea is in this very complicated mathematical diagram on the right. It's essentially very simple, right? We establish an association based on some statistical measure or some machine learning measure. Right now, the framework that we have that Yannick's put together has a number of very simple kind of statistical associations, including things like Pearson correlation. And so our hypothesis is that if two molecules, a protein, are expressed at similar abundances across a large number of conditions, they might be functionally related. So we want to evaluate the extent to which this is true. We know that there's lots of reasons why this might not happen, or lots of cases where this might not happen, but we really believe that under the right kinds of conditions and the way that we look at it across multiple species, that this is actually a reasonable way to look at metabolite function and protein function, depending on which end you have experimental or identification for. So we want to benchmark this. We're benchmarking it first using the KEGG reactions. So the KEGG reactions just capture known enzyme metabolite relationships. And we basically go through our protein and metabolite data that we've gotten experimentally. And we match those up. We say, here's the number of positives that we have where we know that we see the protein and the metabolite. And then for the negatives, we basically just randomize. So we randomize all those connections. So you have a large number of possible connections. We say, if we don't see it in KEGG, it's a negative. It's debatable, but I think it's a reasonable assumption for what we're doing. We have a lot of negatives, and I want you to think about a lot of negative examples when we go into the next slide. And so this is the data slide. And if you can ignore the F1 values that are on the y-axis, but I'll draw your attention to them, they are very, very low. We have a very big false positive problem, and that's partly because we're dealing right now with a very limited amount of data. We've got two different yeasts. These are oleaginous yeasts that we're using for as chassis for synthetic biology, making different kinds of bioproducts. We've got data sets that we've collected and kind of hand data cleaned. One of them has 16 different conditions. Another has 36 different conditions. Some of these are fairly similar kinds of conditions. The range of stuff that we're not getting is not very wide. What I want you to see is when we do this benchmarking and to get the F1 score, which is like the, kind of a mean between the precision and recall, from the individual data sets, the 16 and the 36, they are in the blue and the yellow at the bottom there, and there's really not much signal. If you zoom in and you take out that other green thing, you can actually see that there is a signal. So the thresholding is basically lower correlation on the left, higher correlation on the right. But what I want to point your attention to is the green here, which is our combined. We combine these two data sets, increasing the number of different conditions that we're looking at, but also looking across two different organisms that have different metabolic networks. And we're able to basically start to get rid of some of those false positives and really increase our signal. So this is still very low F1 scores. So what we're doing right now is we need more data. There's a lot of this data around. Turns out that it's a pain in the butt to do the data cleaning. And I think anybody who's dealt with data knows that this is a problem. There's some specific problems with mass spec assisted omics, and metabolomics is a little bit of a wild west. So we're working on that right now, and what we believe is going to be the case is that we're going to be able to continue to improve this. And there's a number of reasons why we think we can talk about it later. So data cleaning is a pain. So really, this is a novel way of associating. Oh, the other thing I wanted to say just for a second is that I've also plotted the traditional methods of doing this on this same plot. You notice there aren't any lines, right? So it's a nice place being where we don't know of any other methods that do this in a formalized way. This is something that's not very it's not terribly innovative on the individual level. A lot of studies that do both proteomics and metabolomics do this kind of on a one-off level. They say, oh, this protein is highly correlated with this metabolic peak, and so therefore we can maybe make some kinds of assumptions or hypotheses about it. But doing it in an organized way with benchmarking, I haven't seen it done. So if you've seen it done, please come and see me. So this is basically the idea. We think that associations will provide insight into unknowns. This is where we can associate, for instance, unidentified metabolic peaks that we might see repeated under multiple experiments with pathways or proteins where we know the function. And vice versa. We might have metabolites where we know something about the function. We can associate them with, for instance, transporters. That's one thing that we really like, we think is going to be a low-hanging fruit, right? Transporters oftentimes are annotated as transporters, and they don't have anything about the substrate. But if we can kind of associate these with substrates, that's going to be hopefully an easy win. And really, this is a complementary approach to the homology-based approaches. So we're thinking once we kind of get this benchmarking done and kind of prove to ourselves and maybe the world that this is something that's worthwhile, we're going to combine these methods and see if we can get more out of it. The other thing that I want to say is that this is, fairly limited in its application. You're not going to be able to apply this to every protein and metabolite. You have to have the data to be able to do it, but we think this is going to be able to give us insights into some unknowns that will be really important. So I just want to thank especially Yannick, who couldn't be here, unfortunately, Lumi Montiero, who's also been working closely on this project. I have a few people who've done some of the experimental work that I didn't really talk about. We've generated some of this experimental data and some interns. And I would take any questions, although I know we're cutting into coffee time. Thank you.
Thank you, Jason. I want to be respectful of people's time, so anybody who wants, now I don't want to stop you, but anybody wants to stick around and ask a couple of questions of Jason, please. Any questions? I have one.
Sure.
Can you go back to your F-score graph? So usually I tell people your F-scores are suspiciously high, yours are suspiciously low. Can you explain how you get to a 0 on these different things? It's kind of
it's not all the way to 0, but it is.
Yeah.
We have close to 100,000 negatives, and we've got like close to 150 positives. So it's a high number of negatives, a dramatically high. So
why not just balance your method?
Because that'd be cheating.
Yeah. Well,
Because with this isn't, we're not training anything. This is actually, we don't have any AI in here. So since we're not training, this is basically just saying, what would we get if we were to take a data set that we already have and apply this benchmarking? How likely is it that we're getting positives? And from this, it's fairly unlikely, but we've got a number of ideas for filtering that we think we can get rid of a lot of those negatives. And I actually have done what you suggest, similar to what you suggested, produced a much more balanced data set, and you get much better results. The balanced data set is if you take only the things that you have positive ends for, and you do the randomization, you get a lot fewer negatives. And if you dramatically reduce the number of negatives, your F-scores look pretty reasonable.
Okay. One more thing about Jason, he draws awesome cartoons. Have you seen there? So if anybody likes to academic laughs, red pen, black pen,
Yeah.
Have fun. Instagram and Blue Sky.
Blue Sky and Facebook.
All right. So see you all after the break, hopefully.